Hello everyone and welcome back to Biology with Mrs. Evans. Today we are looking at the third part of our unit on genetics, which is basically genetic engineering, or also referred to as biotechnology. So to start this topic, we are going to talk about selective breeding. Selective breeding is breeding individuals that have the trait that you want to ensure that you see that trait in your offspring. For example, I have blue eyes. So if I wanted to make sure that I had a baby with blue eyes, then I would choose a male that had blue eyes, and I would basically create a baby with them to ensure that my baby would have blue eyes. We do this all the time in dog breedings to make sure that they have maybe a temperament that we want, a tail set, etc., in order to get those specific traits. So selective breeding, we're not changing the genetic makeup. We're only choosing to mate individuals that have those traits that we want, so that we can ensure those traits are being passed down to the offspring that we are trying to create. Hybridization. Hybridization is crossing dissimilar individuals to bring together the best of those organisms. So you see here the ear kernel on the left has a full kernels on the ear of corn. The kernel here on the right is much bigger, but it doesn't have a full kernel set that you see. So by breeding these two individuals together, you come up with this cross that is basically a hybrid, and you notice that it is much bigger than this one, but it also has the full kernels that you see present here. Hybridization is also the technique that was used to create the breed Doberman Pinschers. Doberman Pinschers is a cross between a Rottweiler, Greyhound, and Chihuahua. They chose the Rottweiler because it had the muscular strength that they wanted in a guard dog. They chose the Greyhound so that it would have the speed of a Greyhound, and then they chose the Chihuahua so that the dog would have the tenacity of the Chihuahua, because if you've ever seen a Chihuahua, they are very small dogs that think that they can basically conquer the world regardless of how big another dog may be. So by choosing those three breeds, those traits that they wanted, and they bred to get the Doberman Pinschers that we have today. Inbreeding, this is something that you are probably already familiar with. This is when two organisms that are closely related, so typically brothers and sisters, fathers and daughters, first cousins, when you breed two individuals that are close together, you see basically an increase in genetic defects. A friend of mine is a breeder of Airedales, and she had a brother and sister that she had, and she was going on vacation, and she put the dogs in a kennel, and she asked the kennel to make sure that the two dogs remained separated because the female was going into heat, and she didn't want to have an accidental litter. She came back, got the dogs. The kennel said that they were kept separate and that there were no issues. And then when she was at work one day, her husband called to tell her that the female was giving birth to puppies. And lo and behold, they were the puppies of basically her brother, and her brother had a really bad cataract in his left eye. That was a genetic condition that was kind of like a recessive trait. But by breeding two brothers and sisters together, all of the offspring were born with really bad cataracts in both eyes, and a lot of the doctors thought that the dogs would be blind because of that. So when you breed two closely related individuals, you increase the likelihood of a genetic disorder or a genetic defect. Genetic engineering. Genetic engineering is making changes to the DNA code of a living organism. So bioluminescence is basically something that we see in jellyfish and in some corals that give them those very bright colors, or being able to glow in the dark, if you will. So by finding that gene, knowing exactly where that gene is, scientists can cut out the gene that codes for that, and then use that gene to basically incorporate that into another organism. So for example, you see here on the screen, they took the gene that causes bioluminescence from this organism here, and then they inserted that into the bacteria, and you notice that the normal bacteria does not have that glow-in-the-dark feature that you see here. But once you inject basically the jellyfish's DNA segment that codes for bioluminescence, you end up with an organism that now has that ability. Restriction enzymes. So restriction enzymes are enzymes that are used to cut DNA at a specific location. Everybody has undoubtedly done a word search before. So you know that you're looking for a specific word and basically this jumble mess of words. But you know what you're looking for, and then once you found it, you simply circle it, and you know that's where the word is. That's exactly how restriction enzymes work. So let's say we have this segment of DNA, and the restriction enzyme is simply looking for the sequence CAT. And the way restriction enzymes work, they find that sequence and then they know exactly to cut the C between a specific base. So let's say this enzyme is looking for the CAT sequence, and it's going to cut between the A and the T. So here's the CAT sequence, and the enzyme will simply come in and cut basically the DNA in this location. And what that creates is it creates two fragments of DNA, one that is 3 base pairs, and then this one over here is 4 base pairs. So by using restriction enzymes, we can create these fragments of DNA of various lengths, and we use that basically to maybe cut out a gene that we want, and I'll talk about how we would get that here in a second. So restriction enzymes are used to cut DNA at specific locations, and then once we've used restriction enzymes, then another technique is basically what's called DNA fingerprinting. So once you have basically exposed DNA to restriction enzymes, what you do is you use what's called gel electrophoresis. Gel electrophoresis is a technique that is used to separate the DNA fragments from one another. So you see here you have basically this is a gel that was created, and in the gel you create these little wells using a comb. And you'll notice that there's some dye there in the gel as well, so that you can see basically the DNA as it moves through the gel. So notice that you have the gel is connected to basically a current. Notice you have a positive side and you have a negative side. And what you do is you take the DNA and you insert it into these wells that you see here. You turn on the current, and DNA is slightly negative, so it is going to move to the positive side of the gel. Opposites attract and like charges repel, so the negative side here is going to be repelling it, so it's going to move through and it's going to be attracted to the positive end. So what happens is remember, restriction enzymes cut DNA at specific locations. You would have exposed the DNA possibly to 3 or more different restriction enzymes, so that you end up with these fragments that are varied in length, and then you would put it in the gel, and what happens is the very small fragments, because they're small, they're going to move through the gel pretty quickly in a certain period of time. So the smaller fragments will end up kind of down here in this part of the gel, and then the larger fragments, because they're so big, they're not going to move through the gel as quickly, so the larger fragments will kind of tend to be here. So as a result of this process, you create a banding pattern basically in the gel, and that is what we call basically a DNA fingerprint. So this is what happens when you basically do electrophoresis. You run a gel. So you would have loaded the DNA, as I said, into the well. Notice that the negative side is basically here, the positive side is here. The DNA is going to run through the gel, all right, and it's going to create this banding pattern that you see here in the gel, and I'll show you what I mean by that in the next slide. So here is basically a template of known DNA fragment sizes. So what you do is you take the DNA sample and you compare it to the fragment. So let's say this fragment here is probably, let's say 20 base pairs. And then you know that this has moved slightly further down than that, so that's probably maybe 18 or 19 base pairs based on where it is compared to the known DNA fragment size. So as I said, this technique is used to create a DNA fingerprint. Notice that you have basically a sample that you would have collected from a crime scene, and then you have 3 suspects that you believe maybe committed the crime, and you do gel electrophoresis to create a fingerprint, and then you compare the two. So if you notice, you have your crime scene DNA, and then you have your 3 suspects, and then you compare the banding pattern that is present. If you notice, suspect 2, their banding pattern is exactly the same as the crime scene. So if you look at it, you'll see they match up. So suspect 2 was definitely at the crime scene and left DNA at the crime scene. So I don't know, what he is, believed to have done, but you can definitely put him at the crime scene based on DNA evidence that you collected at the crime scene. So I talked about using restriction enzymes to cut DNA, and I talked about basically genetic engineering, finding those genes. So the same process would be true for basically if you want to use a gene to create a genetically modified organism. So let's say instead of this being crime scene DNA, that you have basically a gel, and these are the different segments of DNA. I know that the gene that causes bioluminescence, let's say, is going to be found in the gel here. So because it's, let's say, 15 base pairs or whatever. So I run the gel, and then this is the gene that I want. So I simply basically take this segment of DNA out of the gel, and then I use that to incorporate it into another organism. So by using gel electrophoresis, scientists can get the exact gene that they want to see basically in another organism. So let me go back real. So, polymerase chain reaction. This is a process, relatively new process. I think it came available in the 80s. Basically, what this is copying DNA. So, polymerase, that word should be familiar to you. We talked about DNA polymerase and we talked about RNA polymerase. Polymerase, remember, is the enzyme that is going to read the exposed DNA and make copies of it. So the way that polymerase chain reaction works is you take DNA that you want to copy and you expose it to free nucleotides and the enzyme polymerase, and the free nucleotides and the enzyme are going to create lots and lots of copies of DNA. So this is basically showing you the process. You have your DNA that you have collected, you expose it to DNA polymerase, and then the free solution of the nucleotides, like the C's, the G's, the A's, and the T's, and then those free bases that are in the solution and the enzyme are going to be used to make lots and lots of copies of DNA in a short period of time. So DNA polymerase is used to make copies. Polymerase chain reaction is a technique that was used to help make copies of DNA. So DNA evidence, as you probably know, didn't get started or wasn't usable until the 80s, when these processes started to show that they were useful. So one of the things with evidence is that in order for, well, specifically DNA evidence, is that both sides have to have the ability to test it independently of one another. So sometimes DNA is collected in a crime scene, but it's very small amounts. And if there's too small amount for both sides to do the testing, then nothing can be done with that DNA. So DNA was basically being kept in a vault, and nothing was able to be done with it because there was no way that both sides could test it. So polymerase chain reaction was developed by scientists to copy the DNA. So you could take a very small amount of DNA, expose it to basically the polymerase and the free nucleotides, and then you could end up with quantities that now could be tested by both sides. And often there's even some that's left that can be tested 20 years from now if the case gets appealed, etc. So polymerase chain reaction, you need to know, is the process that is used to copy DNA. Manipulating DNA. So again, let's say you have a gene that you want. You can use restriction enzymes to cut out that DNA, and then you can use that piece of DNA and incorporate that into an organism. So that's called recombinant DNA. Basically, DNA that is a mixture of genetic material from two different sources. So hot topic in the news today is genetically modified organisms. Let's say that you have a rose bush that miraculously had the ability to ward off pests, Japanese beetles, whatever. They, because of maybe a chemical that they created, so that when the Japanese beetle bit into the leaf, it didn't like it, so they stayed away from it. So you could find that gene that caused that trait and use that trait or use that gene to basically incorporate it into other roses or maybe even corn or something else. So recombinant DNA is a mixture basically of two sources. So a transgenic organism is an organism basically that has genes from another species. So you take DNA from a coral that has a specific color that you want, and you find that gene that has that color. You get it from the coral's genome, and then you insert it into another organism. So you end up with a organism that would not have that color originally. So this is basically a spider monkey that was used, or they injected basically a glow-in-the-dark that you can see here gene, so that the little spider monkey would glow in the dark. So when I was in school, when you went to the pet store, the only zebrafish that were present were the ones that you see here, these kind of boring zebrafish. So with the takeoff of biotechnology, basically scientists could find the gene that causes these beautiful colors that you see in coral, find that gene, remove it, and then use it to create these beautiful fish that you see here that you would not see in nature, would it not be for basically humans taking the gene that causes this color in jellyfish or this color in coral, finding those genes and then incorporating that into the zebrafish DNA or embryo as it was developing to create these exotic fish that we have in the pet stores today. So again, this is showing you how biotechnology works. So luciferase is a enzyme that causes basically the glow-in-the-dark, or not glow-in-the-dark, the fireflies that we have, or the lightning bugs here, when they basically show that light, is because that enzyme is working. So you find that gene that causes that, you remove it, and then you incorporate that into the tobacco plant that you see here. And what happens is you have a tobacco plant that will now basically turn on and off like a lightning bug or a firefly. Cloning is a technique that you can create a genetically identical copy. So cloning is something that was, I believe it was accomplished for the first time in 1995. This is the actual sheep that they cloned. This is the sheep Dolly. So the way that cloning works is you collect cells from the organism that you want to clone, you remove the nucleus from those cells, and then you take that nucleus and you insert it into an egg. The egg cell, you have removed the nucleus from that cell, and then you insert the nucleus of the organism that you want to clone, and you end up with a exact replica of that organism when it's born. So this is showing you basically what I just said. You have, this was the first organism to be cloned, that was Dolly. You take cells that you have collected, you remove the nucleus that you see here, and then you take the egg cell and you remove the nucleus that you see here. Then you simply insert Dolly's nucleus into the egg cell. You allow the egg to develop into an embryo, and then you take that developing embryo and then you insert it into basically the uterus of a surrogate. And then a couple weeks later, you are born, or the surrogate mother gives birth to basically a exact replica of the animal that you tried to copy. So cloning, you end up with genetically identical organisms. That is basically genetic engineering in the nutshell. Again, this is only a brief overview of all the possibilities that's out there in the world today with genetic engineering. And again, we just highlighted the basics for genetics. If you take this class in college, you'll go into a lot more detail, but these are the things that you need to know. I will see you in class tomorrow. Have a great day. Bye.